There's a fertilized egg drifting somewhere in the open ocean right now. No bottom under it, no surface it needs to reach, nothing around it but water, and the slow blue fade of light going down. It's transparent, which is the ocean's default setting for anything that doesn't want to be eaten. And it's small. Depending on which species we're talking about, it runs somewhere between 7/1000ths and 2/100ths of an inch across, so if it landed on your fingertip, you wouldn't feel it. And unless the light was behind it, you probably wouldn't see it either. That egg is going to become something around 390 feet long. In March of 2020, a remotely operated vehicle called Sebastian was working a canyon off Ningaloo, on the western edge of Australia, launched from a research ship named Falkor. The expedition was led by Nerita Wilson of the Western Australian Museum, and the vehicle was down around 2,070 feet, which is deep enough that the only light in the picture is the light you brought with you. And in that cone of imported light, there was a thing coiled up in a spiral. It wasn't slack, the way a rope goes when you drop it on a deck. The spiral was held open, turning very slowly, like something that had decided the shape was useful and was maintaining it on purpose. The team put lasers on it, which is how you measure anything down there, and the outer ring of the spiral alone came out at roughly 154 feet. That's the ring, just the outside loop. When they worked through the rest of the coil to estimate the full length of the animal, the number they landed on was somewhere near 390 feet. That's an estimate from geometry, not a tape measure laid along the body, because you cannot lay a tape measure along that body without destroying it. But even a cautious reading of that footage puts it among the longest animals anybody has ever recorded. It's a siphonophore, genus Apolemia. And the reason I started with the egg is that the distance between those two things, the invisible speck and the spiral in the dark, is not the kind of growth you're used to thinking about. You grew from a single cell too. Every cell in your body traces back to one fertilized egg. Every one of them carries a copy of that starting genome, and the whole arrangement is uncontroversially one animal. Nobody argues about it. You have a count of one. The siphonophore starts the same way. One egg, one genome, one continuous developmental process with no seams in it. And at the end of that process, you're looking at something that most biologists will describe as thousands of animals stuck together. Same starting material, same uninterrupted development. Completely different answer to the question of how many things are there. Something happens in between. That something is an actual event in the growth of the body, or more accurately, a short series of events, and nothing about it is metaphorical. What I find hard to let go of is how small the events are. There's a shift in where cell division is allowed to happen. There's a shift in how much each finished piece can still do on its own. Those two shifts are apparently enough to move an organism out of the category we'd call an individual and into the category we'd call a colony. So let's watch it happen. In order. From the egg. The egg is already lopsided before anything divides. Work by Christian and Daniele Carre, published in 1993, described fully grown siphonophore oocytes as polarized, with the germinal vesicle, the big nucleus of the immature egg, pushed off to one pole instead of sitting in the middle. Most of the interior volume is taken up by vitelline reserves, yolk, essentially, packed food, because an animal that spawns into open water has no substrate to attach to and no parent to feed from, and the embryo has to run entirely on what it was handed at the start. There's a structure on the outside of the egg called the cupulae, sitting over the polar bodies. Carre and Sardet described it back in 1981 as strongly chemoattractive to sperm. And the part that stuck with me is that the attraction is species-specific. In water where several siphonophore species might be spawning inside the same few cubic feet, the egg has a chemical way of preferring the right sperm. That's a real problem being solved. There are no bodies meeting, no courtship, no coordination beyond timing. The entire mating system is a chemistry problem broadcast into open water, and the egg is doing the discriminating. Fertilization happens, and then the divisions start, and they're odd from the first one. They're unipolar. In a lot of animals, the first cleavage furrow forms as a ring that pinches inward from every direction at once, like a wire tightening around a balloon. In Siphonophores, the furrow starts at one point and sweeps across, like a zipper being pulled. Freeman documented this in 1983, and the Carrés confirmed and extended it. The divisions stay unipolar and roughly equal all the way to the 32-cell stage. After that, the pattern changes. The embryo now has an outer cytoplasmic region holding all the nuclei and an inner region that's essentially the yolk warehouse. And the later divisions stop cutting through the whole sphere. They only affect the outer layer. The interior stays undivided, a shared pantry sitting inside a shell of cells that are multiplying by the hour. Freeman also did something clever and very low-tech. He marked embryos to track the position of that first cleavage furrow, then followed them forward in development. The site of the first furrow turned out to mark the future oral region of the larva, which means the animal's mouth end is being specified at the very first division, before there's anything you could call a body. The same relationship shows up in other hydrozoans, in Clidia, and in Hydractinia. So this isn't a siphonophore quirk. It's an old cyanidarian arrangement that siphonophores inherited along with everything else. Then, gastrulation. And this is where the siphonophore starts to look unlike its relatives. Most animals gastrulate by folding. A sheet of cells buckles inward, or cells migrate in through an opening, and you end up with an inside layer, an outside layer, and a gut cavity. Siphonophores use primary delamination instead. Certain cells in the outer layer divide obliquely or tangentially to the surface, so that one daughter ends up sitting underneath the other. Do that across a whole region, and you've generated an interior sheet of endoderm without folding anything at all. No buckling required. The deck just gets shuffled, but it doesn't happen evenly around the sphere. And that unevenness turns out to be the entire story. The endoderm forms preferentially on one side. That side thickens. And the moment you have a thickened endodermal layer on one side of an embryo and a thin one on the other, you've stopped being radially symmetric. You have a top and a bottom. Biologists call the thickened side ventral. And this is where things get strange for a hydrozoan, because hydrozoan larvae are typically radially symmetric little swimming cigars, and siphonophore larvae are bilaterally symmetric, two-sided. Like us, in that one narrow respect and no other. That thickened ventral endoderm is where every first zooid is going to bud. It's the construction site. Everything that eventually makes this animal a crowd instead of a body gets its permit from that one thickened strip of tissue. How early is that decision locked in? Freeman went after that question directly, working with Mugiea atlantica, a small calicoferan siphonophore common enough in coastal water to be worked with in numbers. He cut embryos in half and watched what each half did. At 6 or 7 hours after fertilization, both halves could produce a normal larva. Whatever the axis was doing at that stage, it could still be regenerated from scratch. By 15 to 17 hours, the ventral endoderm reformed on the cut surface of dorsal fragments, which tells you the tissue still carried the information, but was reorganizing itself around the injury. By 22 to 36 hours, the halves were in trouble. Endoderm formation was delayed. Budding was disrupted. The embryo had lost the ability to talk itself back into a normal plan. So the dorsal ventral axis gets set before gastrulation, stays negotiable for a while, and then hardens. What sets it in the first place, nobody has found. There is no known feature of the unfertilized egg that marks the future ventral side. The molecular machinery is a reasonable guess. WNT, signaling for the oral aberral axis, something in the bone morphogenetic protein family for the dorsal ventral one. based on what those pathways do in Clydia and Hydractinia and in the sea anemone Nematostella. In Siphonophores specifically, that work has barely started. For a group this famous, that's an embarrassing gap, and I think it says something about how hard these animals are to keep alive in a lab, which is a thread that comes back later and comes back hard. The larva that comes out of all this is called a planula, small, covered in beating cilia, bilaterally symmetric, a yolk core, with no mouth yet. And it does not last long. In most species, the planula stage, B, runs under 24 hours before the first zooids start budding. That's a quick turnaround for an animal that's about to assemble something with thousands of parts. Then the oral end starts to differentiate. Yolk reserves get spent. A gastric cavity hollows out in the middle. Gland cells appear at the oral pole. The aboral portion starts producing nematogenic tissue, which is the cell lineage that manufactures stinging capsules, and that tissue is going to be running more or less continuously for the rest of the animal's life. And the thing that emerges from the oral end of that larva is the protozoid, the primary gastrozoid, the first stomach. This is the founding body. All of the swimming bells, all of the feeding polyps, all of the shields and gonads and the various mouthless oddities I'll get to, descend from budding that traces back to this one first polyp and the thickened endoderm sitting underneath it. It's the last piece of the animal that will be built the ordinary way, straight out of embryonic tissue. Everything after this is copies. There's a detail here I keep turning over. In most colonial hydrozoans, the founding polyp is a founder in the political sense, a starting point for a spreading network that goes on to cover a rock and lose track of where it began. In a siphonophore, the founder stays central for life. It doesn't get buried in the crowd, it doesn't get overgrown, and it isn't replaced. The animal keeps its first mouth. Now, the first thing that gets added depends on who the parents were, and that turns out to be a useful window into how the whole group works. Siphonophores come in 3 broad flavors, and the differences are basically about which parts they decided to keep. The cystognaths have a float and no swimming bells at all. That's the Portuguese man-o'-war and its relatives, a small group, only about 5 described species, and they get around by being pushed. The physognaths have both a float and swimming bells, which is the arrangement most people picture when they picture one of these animals. And the chalicophorans have swimming bells and no float, which is by far the biggest group, roughly 170 of the 175 or so species that have been formally described. Hold that in the back of your mind, because it explains why 2 siphonophore larvae sitting side by side in the same jar of seawater can start building completely different things first. In Nanomia bijuga, a physognath and probably the best-studied siphonophore on the planet, the first structure to appear is the float. It starts as an invagination, a small patch of ectodermal cells at a specific position on the larva, and the position is species-specific, folds inward. The invaginated bud pushes down onto the endoderm underneath and forms a roughly circular structure with a cavity inside it. Then the interesting part. The ectoderm lining that cavity starts secreting chitin into it. Chitin is the stuff of insect cuticle and crab shell and fungal cell wall. It's a tough, flexible, waterproof polysaccharide, and it is a very strange material to find in the middle of a jellyfish relative. But if you're going to build a gas-tight chamber inside a body made mostly of water and jelly, you need a liner. And chitin is the liner this lineage had access to. The cells sitting at the base of the bud divide and produce what's called the aeriform ectoderm, which is the tissue that will handle the gas. The whole assembly then gets pushed outward, out past the outline of the larval body, carrying a layer of endoderm with it, and eventually the outside gets covered over by ectoderm as well. The finished float has 2 ectodermal layers with a sheet of chitin sandwiched between them. In some species, the endoderm folds inward to make longitudinal chambers, and the chamber count is a fixed species character, 8 in Nanomia bijuga, 6 in halostema rubrum. Those chambers stay connected to the gastrovascular cavity, which means the float is plumbed into the animal's digestive system. There's no separate circulatory network here. Food canal and buoyancy device share the same pipes. And then there's the gas itself, which is where this stops being merely clever and starts being a little unsettling. The float doesn't fill with air. There's no air down there to fill it with. The animal makes its own gas in a structure called the gas gland. And the gas it makes is carbon monoxide. Jonathan Wittenberg worked this out in the late 1950s and early 60s, tracing the biochemistry in the Portuguese man-o'-war. The substrate turned out to be L-serine, an ordinary amino acid, and the gland carries a striking concentration of folic acid, which is the sort of detail that tells you a specific enzymatic pathway is being run hard rather than something leaking as a byproduct. In the float of Physalia, carbon monoxide typically makes up somewhere between 0.5% and 13% of the gas. And Wittenberg's interpretation was that the carbon monoxide does the initial inflation and then gets slowly swapped out for ordinary atmospheric gases by diffusion and exchange across the float wall. The animal blows the balloon up with one gas and then lets the sea gradually replace it with another. That's at the surface, though, where there's an atmosphere available to trade with. Go down, and the numbers change dramatically. A report published in Science in 1964 analyzed gas from Nanamia Bajuga, floats taken from below 1,000 feet, and the carbon monoxide content was over 90%. There was nothing to trade with down there. The float is running almost pure on what the animal manufactured, and it has to hold that gas against pressure. Depending on depth, the diffusion gradient across the float wall can run to 30 atmospheres or more, which means the gas inside is under constant, relentless incentive to leak out into the surrounding tissue and vanish. The gland has to keep secreting just to stay level. A Siphonophores float is less like a balloon and more like a boat with a slow leak and a very reliable pump. I'll admit I find this one of the most underrated facts in marine biology. Carbon monoxide is the gas that kills people in garages. It binds hemoglobin with roughly 200 times the affinity of oxygen, and it does that in essentially every animal that has hemoglobin. And a lineage of drifting cnidarians looked at that molecule and decided it made a decent lifting gas. They can get away with it because they don't use hemoglobin. There's nothing in a siphonophore for carbon monoxide to poison. An entire class of toxicity simply doesn't apply. And the door that opens is a buoyancy organ nobody else in the ocean can build. So in Nanamia, the float comes first. In halostema rubrum, the float forms on the dorsal side, near the aboral pole, at the same time as the first swimming bell buds on the ventral side. Both at once. In Cortigalma and in Agalma elegans, a larval bract, which is a protective scale I'll come back to, buds ventrally first, and the float forms dorsally afterward. In Forscalia edwardsi, 3 structures appear on the ventral side simultaneously: float, larval bract, and larval tentacle all at once, no waiting. And in the Chalicophorans, the ones with no float in the adult, there's never any float at any stage. The first zooid to bud is the swimming bell, almost without exception, and it almost always buds on the ventral side. Below it, the primary tentacle develops. Then the oral end of the planula differentiates into the protozooid, and the animal is off and running with no buoyancy organ at all, which raises the obvious question of how a Chalicophoran keeps from sinking. The answer involves ion chemistry rather than gas, and I'll get to it because it's one of my favorite pieces of engineering in the whole group. Now, the swimming bell, the nectophore. I want to be precise about what this thing is, because the standard description undersells it badly. A nectophore is usually introduced as a swimming bell, a propulsion unit, a muscular sac that squirts water. All true, But that's a description of a job, and the job doesn't tell you what the object is. Watch how it develops. Budding starts with a delamination of ectoderm, which is the same trick used in gastrulation. The newly produced cells push down onto the ventral endoderm and proliferate, and what they form is called the entocodon, the medusa nodule. If you know your cnidarian development, that word just gave the whole thing away. The entocodon is the structure that forms a jellyfish. In a typical hydrozoan life cycle, you have a polyp stage attached to the bottom, and the polyp buds off medusae, and the medusae swim away, mature, spawn, and produce larvae that settle and become polyps again. The entocodon is the cell mass inside a medusa bud that hollows out to become the subumbellar cavity, the space under the bell. It's the specific developmental signature of jellyfish formation. So the entocodon appears, and it grows, and it hollows out, and it projects away from the larval body, with its long axis running perpendicular to the oral aboral axis of the planula. Then asymmetric growth swings it around until the long axis is parallel instead. Christian Carre described this rotation in the 1960s, and it's a small piece of choreography that matters a lot, because the direction that bell ends up pointing determines the direction the animal will travel for the rest of its life. Meanwhile, the ventral endoderm underneath hollows out into gastrovascular canals, and one of those canals extends into a structure called the somatocyst. The bell's musculature differentiates. Circular muscle, mostly, laid down in sheets, because a jet is produced by squeezing a volume of water out through a narrow aperture, and that requires a muscle that contracts inward from all sides. And then it stops. It doesn't detach. It doesn't develop a mouth. In most species, it never develops tentacles, or sense organs, or gonads. It stays exactly where it budded, wired into the parent, and it spends its entire existence pumping. That's a jellyfish. Structurally, developmentally, by every criterion of homology anybody has been able to apply, that swimming bell is a medusa. A medusa that was built by the standard process and then never released. And the same logic runs through the whole colony. The gonophores, the reproductive zooids, are also reduced medusae, some of them so reduced they're little more than a sac of gametes with a vestigial bell around them. The feeding polyps are polyps in the full technical sense, with mouths and tentacles and everything. What a siphonophore has assembled is a body made out of animals that in any related lineage would have gone off and lived on their own. That's the sentence people usually reach for, many animals pretending to be one. And I understand why it gets used, but I'd like to push back on the word pretending, because nothing here is pretending. A nectophore that never leaves is a jellyfish whose development was truncated at a specific step, and truncated in exactly the same way, in exactly the same position, in every colony of that species that has ever existed. There's no acting involved anywhere in it. What there is construction, repeated identically every single time. There's one more piece of the larval phase worth flagging, and it's the sort of thing that only makes sense once you know it's coming. Some of these early zooids are temporary. The larval bract, the larval tentacle, the first nectophore in certain families, they get built. They do a job during the period when the animal is small and vulnerable, and then they're shed. The technical word is catechus, which comes from the Latin for falling, and it's the same word botanists use for leaves that drop. Larval nectophores are retained for life in the spheronectids and praids. In the diphyids, they get replaced by definitive nectophores built to a different specification, and you can tell them apart under a microscope by shape. Larval bracts persist in the agalmatide physonex and fall away in a lot of other groups. So before the animal is a day or two old, it already has a scaffolding it intends to discard. Baby teeth, more or less, except the baby teeth are entire small animals. The transition out of the larval phase gets its own name. In physonex and cystonex, the post-larval form is called a siphonula. In Calicophorans, it's a caliconula. Different names for the same threshold, the point where the animal stops budding individual zooids one at a time out of embryonic tissue and starts running a production line. Because that's what comes next. And it's the single most important structure in the entire animal. The body starts to stretch. Between the float at one end and the first feeding polyp at the other, a length of tissue begins elongating. This is the stem, and it's going to be the spine of everything that follows. Though calling it a spine is misleading because there's nothing rigid about it. It's a hollow, muscular tube. The gastrovascular cavity runs all the way through it, so the stem is simultaneously the animal's skeleton, its gut, its circulatory system, and its highway. Food digested in a mouth at one end can travel to tissue 300 feet away, and there's no heart anywhere in the system. Movement through those canals is driven by cilia and by the contraction of the stem itself. As that stem elongates, something separates. In the siphonula stage, the region that was doing all the budding splits into two regions, and the elongating stem carries them apart from each other. From that moment on, the animal has two construction sites instead of one, and they build different things. The forward one sits just behind the float. It's called the nectosomal growth zone, and it makes swimming bells, only swimming bells. The region of stem it fills up is the nectosome, and in a mature nanomya, that's a dense, tightly packed column of nectophores arranged in two opposing rows, overlapping each other like roof tiles. The rear one sits just behind the last swimming bell at the front end of the region called the siphonosome. It makes everything else: feeding polyps, tentacles, protective shields, mouthless auxiliary polyps, reproductive zooids, the whole catalog. That's the architecture. A float, a bank of engines, then a long trailing body of working parts, with a growth zone at the top of each of the two sections, quietly manufacturing. I want to slow down on those growth zones, because everything that makes a siphonophore weird traces back to them. Each one has a structure at its anterior tip that Stefan Siebert, Casey Dunn, and their colleagues called the horn. It's a protrusion, a small bulge of tissue sticking out of the front of the growth zone, and it is the only place in the entire animal where new bodies come from. Every zooid the colony will ever own gets manufactured there, without exception. They demonstrated this in a paper published in 2015, tracking interstitial stem cells through the colony. Interstitial stem cells, I cells, are the general purpose cell type of cnidarians. They're the reason a hydra can be chopped into pieces and rebuild itself, the reason a lot of colonial hydrozoans can regenerate more or less anything. They're multipotent. Give an eye cell the right signals, and it can become a nerve cell, a gland cell, a stinging cell, a gamete. In most colonial hydrozoans, eye cells are distributed. They sit throughout the stolons, the runners connecting the polyps, spread out along the whole organism like a maintenance crew on permanent standby. Damage happens somewhere, and there's a local supply of undifferentiated cells nearby that can take care of it. That's not the arrangement in a siphonophore. Siebert and colleagues found eye cells concentrated at the tip of the horn. They found them in the youngest zooid buds, and as they followed zooids down the stem from young to old, they watched the eye cell population get progressively restricted, confined to smaller and smaller regions inside each zooid, until in the oldest zooids, they were essentially gone. And then, the finding that reframes the whole animal: no eye cells in the stem between maturing zooids. The connective tissue linking the parts of a siphonophore together, the thing that makes it a continuous organism at all, has no developmental potential in it whatsoever. It's plumbing and muscle, nothing else. So the animal has centralized its ability to build, all of it, into a bulge of tissue a fraction of a millimeter across, sitting at the front of each of two regions, and everything that will ever be part of that colony has to come out of one of those two spots. Now, how does a single bud become a whole functional cluster of different organs? It doesn't, in the sense you'd expect. A bud forms at the tip of the horn, and at that stage it isn't any particular kind of zooid yet. It's a probud. As the stem keeps elongating and the probud gets carried backward away from the horn, it subdivides. It parcels itself out into several separate buds, and those buds differentiate into the different zooid types in a fixed order, in a fixed arrangement. The finished cluster is called a chormidium. It's the repeating unit of a siphonophore body, and depending on the species, it might contain a feeding polyp, its tentacle, one or more mouthless polyps, a protective shield, and a set of reproductive zooids, all arranged in a pattern so consistent that taxonomists use the arrangement to tell species apart. The process is called probud subdivision, and Casey Dunn and Gunnar Wagner argued in 2006 that it's a shared derived feature of the Cotonophora, the group containing physonects and chalicophorans. It's an innovation, and it lines up with the origin of the chormidium as an integrated unit, rather than a loose assortment of zooids budding independently. Their argument is that this is what let siphonophores diversify the way they did. Once the whole functional module comes out of a single bud as a package, you can start modifying the package, which means the manufacturing sequence goes like this. Stem elongates, probud forms at the horn, probud is carried backward, probud divides, subbuds differentiate, chormidium matures. Repeat, repeat, repeat for as long as the animal is alive and fed. And that produces a consequence that I don't think gets appreciated enough. Position along the stem is age. The zooids closest to the growth zone are the youngest ones, the ones that budded most recently. The ones farthest away are the oldest. There's no shuffling, no migration, no reorganization. A zooid buds where it buds, gets carried backward by the growth of everything in front of it, and stays in that spot for the rest of its existence. The colony is a conveyor belt where nothing ever gets taken off, which means that when you look at a siphonophore, you're looking at a complete developmental time series laid out in space. Think about what that's worth. Normally, to study how an organ forms, you need a lot of animals at different stages, or time-lapse imaging over days, or a way of freezing development at chosen moments. In a siphonophore, one specimen gives you the entire sequence at once. Right at the horn, the first faint bulge of a bud. An inch back, the same structure a little further along. Another inch, further still. All the way down to a fully mature working zooid at the far end. It's a flipbook that got unbound and laid out flat, every frame sitting next to its neighbors, the whole animation visible in a single glance. The film is still running at one end. The frames it already printed are still hanging there behind it, unchanged, in order. That single property is a large part of why Nanomia bejuga turned into a model organism. Nobody chose it for convenience, and I'll be blunt, it is not an easy animal to keep alive. They chose it because it does your developmental staging for you. But that same property has a cost, and the cost is the beginning of the answer we're heading toward. Think again about where the eye cells are. Concentrated at two horns, dwindling as zooids age, absent from the stem entirely. Now imagine something takes a bite out of the middle of the colony. A fish snaps at it, or a net drags across it, and you lose a section of stem with a dozen zooids in it. There is no local repair crew. There's no reservoir of undifferentiated cells sitting in that neighborhood waiting to be told what to do. The growth zone is far away at the front, and it doesn't move. And it doesn't insert new material into the middle of the sequence. It only adds to the front of the cyphosm and pushes everything else backward. So the wound closes. The animal survives usually, but that section of the body is gone permanently, and every part downstream of the break, if the break is complete, is now disconnected from the only mouth-bearing engine of new construction that the colony has. The pieces of a siphonophore are not, in general, capable of rebuilding a siphonophore. That sounds like a limitation, and it is. It's also the single most useful diagnostic anybody has for the question of what counts as one animal. Because if you cut a hydra in half, you get 2 hydras. If you cut a planarian into a dozen pieces, you get a dozen planarians. Those animals fail the test of individuality in a specific way. Their parts retain the capacity to be wholes. A siphonophore's parts mostly don't. That's a developmental fact, not a philosophical one, and it's measurable. It's the location of a cell population. One more thing about the growth zones before we start looking at what they produce. They don't stop. There's no adult size that a siphonophore grows to and then maintains. As long as the animal is fed and undamaged, the horn keeps making probuds, and the stem keeps elongating and chormidia keep accumulating at the back. Growth is open-ended, indeterminate, in the technical term. That's why the length question in these animals gets so slippery. When somebody asks how big a siphonophore gets, the honest answer is that it depends how long it's been alive, how much it's been eating, and how recently something bit a piece out of it. There's no target. The animal is a process that hasn't stopped yet, and the number you get is a measurement of that process caught at a moment. That's how you get from an egg you can't see to a spiral longer than a blue whale, incidentally, without any of the scaling problems that limit ordinary bodies. A whale getting bigger has to solve 100 simultaneous engineering problems because doubling a length cubes a volume and squares a cross-section, and the bones and the heart and the metabolism all have to be renegotiated. A siphonophore getting longer solves nothing because it isn't making anything bigger. It's just running the same small machine for more cycles. The parts stay the same size. There are only more of them. So what actually comes out of that probud? Let's take the chormidium apart, one zooid type at a time, and pay attention to what each one can do and what each one can't. That second half is the part that matters later. Start with the gastrozoid, because everything else in the colony is downstream of it in the most literal sense. A gastrozoid is a feeding polyp, and it looks like one. There's a mouth at the tip, sitting on a raised cone called a hypostome, and the mouth opens into a gastric cavity, and that cavity connects into the stem canal. Around the base, there's a swollen region called the basigaster, and the basigaster is a factory. It's packed with cells producing nematocysts, the stinging capsules, which then migrate out to where they're needed. And attached at that base is a single tentacle, one mouth, one tentacle, per gastrozoid. That's the pattern. These are the only parts of a siphonophore with mouths. In a colony with, say, a few hundred chormidia, there are a few hundred openings into the animal, and every calorie that colony ever acquires comes in through one of them. Prey gets caught, hauled in, engulfed by a gastrozoid that can distend impressively to swallow things wider than itself, and digested. Then the products go into the gastrovascular canal system and get moved around the body by ciliary currents and by the muscular contraction of the stem, which means the swimming bells at the front, which have no mouths, are being fed by feeding polyps that may be many feet behind them. The gonads are being fed the same way. So is the growth zone, which is doing the most metabolically expensive work in the animal and has no way at all to acquire its own food. It's an economy with one currency and one distribution network and no storage to speak of. Next, the palpon. A palpon looks like a gastrozoid that lost an argument. Same basic polyp shape, similar tissue layers but no mouth. It's a closed sac. And instead of a full tentacle, it carries a thin reduced filament called a palpicle, which in some species retains stinging cells and in others doesn't. What is it for? The honest answer is that the picture is incomplete, and I'd rather say so than fill the gap with confidence I don't have. The best supported roles are circulatory and excretory. A palpon is plumbed into the stem canal. It can contract, and contracting a blind sac attached to a canal system pushes fluid around. There's good reason to think palpons help drive circulation through the colony. There's also evidence pointing at waste handling, and in some groups they clearly take on a defensive role, loaded with stinging cells, positioned around the vulnerable parts. What's not in dispute is where the palpon came from. It's a modified gastrozoid. Take a feeding polyp, delete the mouth, reduce the tentacle, and you have one. In development, it buds from the same probud in the same cluster as the gastrozoid it's related to. The colony is producing variations on a single polyp theme by adjusting what gets built and what gets skipped. Then there's the bract, and the bract is the odd one out. Every other zooid type in a siphonophore has an obvious counterpart elsewhere in the cnidarian world. Polyps look like polyps, medusae look like medusae. The bract looks like nothing else. It's a gelatinous scale, often thick, often shaped a bit like a wedge or a shield or a helmet depending on the species, and it sits over the delicate zooids in the cormidium the way a roof tile sits over a rafter. There's no consensus on what it's homologous to. It's a structure siphonophores invented and nobody else has. Its first job is armor, and jelly armor works better than you'd think against the things that eat you in open water when you weigh almost nothing. Its second job is buoyancy, and the mechanism is one of my favorite bits of chemistry in the ocean. Seawater contains sulfate ions. Sulfate is heavy. The mesoglea of a jellyfish, or a siphonophore, is mostly water with dissolved salts in it, and if the salt composition matches seawater exactly, then the tissue is neutrally buoyant at best. But if you can selectively pump the heavy ions out and let lighter ones in to maintain the charge balance, the tissue becomes less dense than the water around it. It floats. That's what bracts do. Measurements on siphonophore bracts have found something on the order of 44% of the heavy sulfate ions in the mesoglea replaced by lighter chloride ions. The animal is running an ion pump to make itself buoyant, and in some calicophorans, the ones that never build a float at all, this appears to be adjustable along the length of the colony, so different regions can be tuned to different densities. Think about what that solves. A calicophoran has no gas float, no swim bladder, no oil reserve, no fat. It stays where it is in the water column by controlling the salt chemistry of its own jelly. And it does that in structures that are, developmentally, individual zooids. In these animals, buoyancy is a job, and the job is held by a body part that also happens to be an organism. Bracts have a canal running into them, the bracteal canal, connected to the same gastrovascular system as everything else. So even the shield is plumbed in. Even the roof tile has to be fed. Then, the gonophores. These are the reproductive zooids, and like the swimming bells, they're medusae, reduced ones. In some species, a gonophore is recognizably a tiny jellyfish with a bell and radial canals and everything just permanently attached and never released. In others, the reduction has gone much further, until what's left is essentially a sac of developing gametes with the vestigial architecture of a medusa wrapped around it. They come in male and female versions, and in many species, including Nanomia, a single colony carries both, which means a siphonophore colony can be simultaneously a male and a female, made of many small males and many small females, all of them clones of each other, and of the swimming bells, and of the stomachs. When they're ready, gametes are released into the water. And that's the moment the whole story loops back to the first thing I described. Those gametes drift. They find each other by chemistry, and one of them becomes an egg with a cupule on it that starts sweeping through unipolar cleavages. Sexual reproduction is the only point in the entire life cycle where a siphonophore produces something with a new genome. Everything else, every zooid, every bell, every bract, every stomach across 300 feet of animal, is asexual budding. One genotype, thousands of bodies. Now let me raise the vocabulary problem because at this point it's unavoidable. I've been calling these things zooids and using words like organ and body part, and the two categories don't fit together. An organ is a piece of an animal. A zooid is an animal. Both descriptions apply here, and each one is defensible with real evidence, and neither is complete. The evidence for organ goes like this. Zooid types are functionally specialized to a degree you don't see anywhere else in the colonial animal world. They're arranged in precise, species-specific patterns, rather than growing wherever there's room. They can't survive detached. They're physiologically integrated through a shared gut. Work published by Siebert, Dunn, and colleagues comparing gene expression across Siphonophore species and across zooid types found that zooid types carry characteristic expression profiles, which is exactly what you'd predict if you were looking at differentiated organs rather than at copies of a whole animal. The evidence for animal is just as solid. Every zooid develops through the standard developmental route of a free-living polyp or a free-living medusa, using the same structures at the same stages, right up until the point where the process stops early. The swimming bell builds an entocodon. The gonophore builds an entocodon. Each of them is a truncated animal that followed the ordinary route and halted partway. Both readings are correct, which usually means the question is badly posed. And I'll say plainly that I think the word colony does more harm than good here. Colony imports a picture of independent citizens who chose to live together, which is roughly right for a coral head and roughly wrong for a siphonophore. A coral polyp can feed itself, defend itself, and in many cases survive if its neighbors die. A siphonophore nectophore cannot eat. It has no mouth. It never had one, it never will. And separated from the stem, it will burn through whatever's left in its tissue and stop. The load-bearing point is this one. Look at what each part can't do. The nectophore swims and cannot eat, cannot reproduce, cannot sting. The gastrozoid eats and cannot swim, cannot reproduce, and would drift helplessly if released. The palpon can't even eat. It has no mouth. The bract has no mouth, no muscle worth speaking of, no nervous system to speak of, and does nothing but shield and float. The gonophore reproduces and does nothing else. Every single part of this animal is missing something essential, and every missing piece is covered by a different part. That's not cooperation between organisms. That's a body with the division of labor pushed to a place no other animal has taken it. And the crucial thing, the thing that ties this back to development, is that none of it was negotiated. There was no allocation of roles. What determined whether a bud became a stomach, or a shield, or a gonad, was which sub-bud of the probud it happened to be, and where in the chormidium that sub-bud sat. Position at the moment of subdivision decided the entire future of that body, and since nothing migrates and nothing rearranges, the decision was final within hours of the bud appearing. Place is destiny in this animal, to a degree that's almost uncomfortable. There's a nice illustration of how far the tinkering can go, and it comes from the most famous siphonophore of all. The Portuguese man-o'-war has a structure that had confused people for a long time. It carries the long fishing tentacles everyone knows about, the ones that can trail for many yards under the float, and the question was, which zooid those belong to. Katrina Monroe, Casey Dunn, and colleagues published a study in 2019 using optical projection tomography on larval and juvenile specimens, essentially building three-dimensional reconstructions of the developing colony, and they tracked the budding order directly. What they found is that the tentacle-bearing zooid and the feeding zooid come from separate buds. They're different individuals, and the tentacle bearer is a modified gastrozoid with an enlarged tentacle, no mouth, and a specialization for producing stinging cells. They proposed calling it a tentacular palpon and argued it's unique to this species. So the Portuguese man-o'-war has taken a feeding polyp, removed its mouth, blown up its tentacle to enormous length, and converted it into a dedicated weapons platform. The thing that stings you at the beach is a stomach that gave up eating in order to specialize in fishing, permanently attached to a relative that kept its mouth and does the swallowing. That's what pro-bud subdivision buys you. Once a whole functional module comes out of one bud as a unit, evolution can start editing the module. Delete a mouth here, extend a tentacle there, duplicate this part, suppress that one. The result is a group of animals that solved specialization the way an engineer would by making dedicated components, except each component is homologous to a jellyfish. All right, the animal is now a few weeks old in the case of something like Nanomia, and it has a float, a column of swimming bells, and a trailing body of chormidia. Let's watch it move. The nectosum, that packed column of bells behind the float, is arranged in two opposing rows. Each bell overlaps its neighbors, angled, nested, so the whole column looks a bit like a pine cone or a braid depending on the species and the angle you're looking from. Every one of those bells is a working jet engine. The mechanism is old and simple. Water fills the space under the bell. Circular muscle sheets contract, squeezing the bell inward. Water is forced out through a narrow aperture, and the reaction pushes the animal the other way. That aperture is ringed by a muscular flap called the velum, and the velum is more than a nozzle. It can be deformed. Contract one side of it, and the jet comes out at an angle, which means the bell can vector its own thrust without changing its own orientation. So far, so jellyfish. What makes the Siphonophore version remarkable is the fleet. In 2015, John Costello, Sean Collin, Brad Gemmell, Kelly Sutherland, and John Dabiri published a study in Nature Communications that measured what the individual bells of Nanomia bejuga were doing during real swimming. They tracked the jets. They measured the angle each jet came out at relative to the axis of the animal, and they compared bells at different positions along the nectosome. The bells at the front, the young ones nearest the growth zone, were firing their jets at angles closer to perpendicular to the body axis. The bells at the back, the older, larger, more powerful ones, were firing more nearly parallel to it. Perpendicular thrust at the front of a long body is a steering input. It's torque. It swings the nose. Parallel style thrust anywhere is forward propulsion, and the biggest bells were doing the most of it. So the animal has a division of labor in its engine room. Turning is handled by the small young bells up front, which have the worst power output and the best leverage. Forward drive is handled by the big old bells at the back, which have the power and no leverage worth using. And the two jobs are physically separated along the length of the animal. The part I keep coming back to is this. Nobody assigned those jobs. Remember where new swimming bells come from? The nectosomal growth zone, at the very front just behind the float. Every new bell is inserted at the leading end of the column, which pushes the whole existing column backward by one position. A bell is born at the tip. It's small, it's weak, and it's sitting at the point of maximum leverage. Over time, as more bells are added ahead of it, it gets carried backward. And as it's carried backward, it grows. It gets bigger, stronger, and less useful for steering, and it slides into a position where its power is worth more than its leverage. The job description changes because the position changes, and the position changes because of budding. And the budding is happening in a bulge of tissue that has no idea what any of this is for. The result is a propulsion system where the youngest components steer and the oldest components push, and the transition between roles is gradual and automatic and requires no decision-making of any kind. Costello and colleagues put it in terms of clonal development organizing multi-jet propulsion, and I don't think there's a better one-line description of what Siphonophores do than that. Order of manufacture produces functional architecture. Now, does it actually work? A follow-up study measured maneuvering performance directly, and the numbers are better than I expected. The animal turns by firing a single bell at the apex, one engine, and the resulting length-specific turning radius averaged about 1/7 of its own body length. Mean angular velocity ran a bit over 100 degrees per second, with maxima above 200, and the fastest turn they recorded came in at 363 degrees per second. That's a full rotation and then some in 1 second by an animal with no brain. For scale, that turning performance is in the range of squid and boxfish, and it beats a lot of vertebrates outright. Then there's the reversal, and the reversal is the result that made me sit up. Nanomia swims backward at essentially the same speed it swims forward. Both directions came in around 134 millimeters per second, which is a bit over 5 inches a second, or about 37 times the length of a single swimming bell every second. During those reversals, the jets themselves were clocked above 1,000 millimeters per second, roughly 3.5 feet per second. Coming out of a bell, you could balance on your fingernail. A 1-to-1 ratio of forward to reverse speed has not been recorded in any other swimming animal. Fish are worse in reverse. Squid are worse in one direction than the other. Almost every swimmer on Earth has a preferred end. This one doesn't, and the reason it doesn't is that the velum can vector the jet through a wide enough arc that the same engines work either way. During a reversal, all the bells fire together, all vectoring at once. Think about what that's worth to an animal shaped like a string with a fragile fishing apparatus hanging off the back. If something threatens the front, you don't want to turn around. Turning around means swinging 300 feet of tentacles through the threat. You want to be somewhere else immediately, and the fastest way out is straight back the way you came, which raises the question of how the bells know to fire together. There's no brain here. There's no ganglion, no central processor, nothing that could be pointed at as the place where the decision gets made. But the coordination is unmistakable. All the bells fire together for a reversal. A single apical bell fires alone for a turn. Something is routing signals. George Mackey spent decades on this problem, starting in the 1960s, and the picture that emerged is a system built for speed rather than for computation. Running through the stem of Nanomia, there are giant axons, 2 of them. Giant axons are the classic solution to the problem of conducting a signal fast down a long body, and they're famous from squid and from certain polychaete worms, where they drive escape reflexes. Finding them in a cnidarian was a surprise when it happened. An action potential in one of those giant axons sweeps excitation along the whole length of the stem in a hurry. But the axons don't connect directly to the individual swimming bells. Instead, the excitation feeds into a diffuse polygonal nerve net that's distributed through the stem, and the nerve net talks to the bells. There's also a separate set of nerve tracts, identified by their reactivity to an antibody against a small neuropeptide called FMR-famide, and those tracts link specific bells on opposite sides of the nectosome to each other. More recent work on the stem nervous system in Nanomia has traced 2 distinct routes into a swimming bell. One of them triggers an epithelial impulse, a wave of electrical activity conducted through the sheet of skin cells themselves rather than through neurons, and that route produces reverse swimming. The other produces forward swimming. Same bell, 2 input lines, 2 behaviors, so the architecture is a fast trunk line, a distributed local network, and a small set of dedicated cross connections. That's a nervous system designed around the problem of getting many separate bodies to act simultaneously, which is a different design problem from the one our nervous systems solve. We have a central controller sending instructions to muscles that were always parts of us. A siphonophore has a broadcast system reaching a fleet of individually complete swimming animals that need to agree on a direction. And it doesn't always work perfectly, which I find reassuring. Observations of these animals include plenty of uncoordinated firing, bells going off out of sequence, colonies wobbling and correcting. The integration is real, and it's also imperfect, which is roughly what you'd expect from a body that has to keep re-recruiting new members into the fleet every few days. There's a preparation step too, and it's easy to overlook. Before a siphonophore can do anything athletic, it has to deal with its own fishing gear. A feeding colony has tentacles spread across a large volume of water. You cannot accelerate through that arrangement. So the stem contracts hard and fast, hauling the tentacles in against the body, and only then does the animal move. Watch ROV footage of one of these being approached by a vehicle, and you'll see it. A fast, shocking contraction that pulls the whole spread-out structure into a compact bundle, followed by the jets. It's the same instinct as a fisherman reeling in before starting the engine, and it costs time. And that lost time is probably one of the more important constraints on how these animals live. Now put all this in motion over a day. Nanomaya bijuga is a vertical migrator, and a serious one. In Monterey Bay, where MBARI has run quantitative video surveys for decades, the population sits mostly between about 650 and 1,300 feet during daylight, with the full range running from around 30 feet down to half a mile. At night, a large part of that population rises toward the surface to feed, and before dawn it goes back down. That's a commute of hundreds of feet, twice a day, every day, for an animal a few inches to a couple of feet long. Powered by a column of bells you'd need a hand lens to count. And it's doing that commute while carrying a sealed gas chamber. Pressure changes with depth. Gas volume responds to pressure. Any animal hauling a fixed gas-filled space up and down through hundreds of feet of water column has a physics problem to manage, and the float has to hold its contents against a diffusion gradient that gets more punishing the deeper it goes. The gas gland doesn't get to rest. None of this required a decision. The migration is driven by light, the prey field moves for its own reasons, the animal follows. But sitting inside that routine is a body whose steering is determined by budding order, whose buoyancy is determined by ion pumps and gelatinous shields, whose signals travel down two giant axons into a nerve net, and whose engines were medusae that never got to leave. And it still hasn't eaten anything in this story. That's next, and the feeding apparatus is where these animals stop being merely strange and start being frightening. A siphonophore hunting looks like nothing at all is happening. The jets stop. The animal hangs there, oriented however it wants to be oriented, and the stem relaxes. And out of every feeding polyp along the body, a tentacle unfurls and drifts outward. And each tentacle carries dozens or hundreds of side branches, and each side branch unrolls in turn. What builds out of that is a curtain. In some species, it's an actual curtain, a broad sheet of near-invisible threads spread across the water. In others, it's a cone, or a cylinder, or in the case of the apolemia off Ningaloo, a slow, open spiral with the whole colony forming the coil and the tentacles filling the space inside it. The strategy is ambush by volume. Chasing prey in open water is expensive, and it's especially expensive for an animal with no eyes, no speed to speak of, and a body that would be shredded by any real turbulence. So the siphonophore does the opposite. It converts a very small amount of tissue into a very large intercepting surface, and waits for the ocean to move things through it. I want to give you a sense of how good that trade is. A colony a few feet long, weighing about as much as a soaked paper napkin, can hold a fishing structure spanning a volume you could park a car in. The tissue investment is negligible. The energy cost of holding it there is close to zero, because most of the structure is neutrally buoyant jelly, and the animal isn't swimming. The only real expense is the manufacture of the weapons, and those get built continuously in the basogaster of every feeding polyp, which is doing that job anyway. Now let's look at what's actually on those threads, because the branch coming off a siphonophore tentacle is one of the more elaborate structures any animal has built at that size. It's called a tentillum. The plural is tentilla. And in the more derived groups, it has 5 recognizable parts, At the base, there's the pedicle, a flexible stalk connecting the tentillum to the main tentacle. Then the cnidoband, which is the business end, a band of tissue derived from the epidermis, coiled up tight, absolutely loaded with stinging capsules. Running alongside the cnidoband, attached to it, there's a rigid strand made of collagen derived from the mesoglia. It's called the elastic strand, which Alejandro Damian Serrano and colleagues have noted is a bit of a misnomer, since it isn't especially elastic. It runs up the length of the cnidoband and back down again. At the far end, there's the terminal filament, tightly coiled, sensitive to stretch, carrying a different class of stinging capsules that work by adhesion. And wrapping over the whole assembly, there's the involucrum, a sheet of epithelium that grows out from the pedicle and in some species covers the cnidoband completely, like a sheath over a folded blade. Now watch what happens when a copepod swims into it. The first contact is with the terminal filament. That filament is coated with two kinds of capsule, demonums and rhopalonums, and both of them work by sticking rather than by piercing. Desmonemes fire a coiled thread that wraps around a bristle or a hair and grips it. So the prey isn't stabbed at this stage, it's glued. That contact is the trigger. And the terminal filament is stretch sensitive, so the prey's own struggling is what fires the main charge. Then the tentillum unloads. The distal end of the cnidoband shoots outward. The proximal end detaches from the pedicle and slings forward. The coiled band uncoils and whips across the prey, and as it goes, the stinging capsules along it fire in sequence, each one discharging as it comes into contact with the surface. Penetrating capsules called heteronemes drive threads into the animal. Entangling capsules called haplonemes wrap and hold. And the collagen strand, the one that isn't very elastic, stays attached at both ends and becomes the line, the physical connection between the tentacle and whatever is now stuck to it, so the prey can be hauled in. The whole thing is a spring-loaded harpoon and net launched off a thread thinner than a hair. Damian Serrano and colleagues filmed these discharges at high speed and measured how fast the cnidoband travels. In the Euphysinax, the group containing the larger Physanax siphonophores, the average discharge speed was around 225 millimeters per second, which is roughly 9 inches a second. In Chalicophorans, it was much slower, around 1.5 inches a second. And discharge speed turned out to be strongly correlated with the length of the cnidoband, which makes mechanical sense. A longer coil stores more and unwinds further. Within a single tentillum, the different capsule types don't fire at the same rate either. Haplonemes discharge nearly 3 times faster than heteronemes. The entangling capsules get there first, and the penetrating ones follow, which is exactly the order you'd want if the goal is to hold the prey still long enough to hit it properly. For a sense of the timescales inside the individual capsule, the best measurements come from a freshwater relative rather than from a siphonophore. Work on Hydra timed the discharge of a single nematocyst in the range of hundreds of nanoseconds. That's among the fastest movements known in any living cell, driven by osmotic pressure inside the capsule that had been building since the capsule was manufactured. It's stored energy released by a trigger, the same principle as a mousetrap, at a scale where the whole apparatus fits inside one cell. So why is any of this necessary? Why not just have a sticky tentacle? Because of what siphonophores mostly eat. The single most abundant animal group in the ocean is the copepods, small crustaceans, 1 millimeter or 2 long, and there are unfathomable numbers of them. They're also extremely difficult to catch. A copepod is covered in mechanoreceptor bristles that detect fluid deformation in the water around it. And when those bristles register an approaching disturbance, the animal launches itself away in a jump measured in milliseconds. Relative to its size, a copepod escape is one of the most explosive accelerations in the animal kingdom. That sets two hard constraints for anything trying to eat them. Your capture surface has to be hydrodynamically quiet, meaning it can't push water ahead of it, because pushing water is exactly what a copepod is listening for. And your strike has to be faster than the escape. A tentacle spread motionless in the water and made of nearly transparent tissue at nearly the density of seawater is about as quiet as a structure can get. And a spring-loaded coil that releases stored energy is one of the few ways to get a fast strike out of an animal with no fast muscle. The siphonophore solution is to be invisible until touched, and then to be very fast for a very short distance. That's the whole design. And the design is tuned, species by species, to the target. Damian Serrano, Stephen Haddock, and Casey Dunn published a study in the Proceedings of the National Academy of Sciences, in which they measured 29 morphological characters of tentilla across 45 siphonophore species, mapped those measurements onto a phylogenetic tree, and tested them against what each species is known to eat. The associations came out clearly. Species that specialize in fish carry more penetrating capsules per tentillum, and the capsules themselves are larger. The entangling and adhesive capsules get rounder in shape. The small adhesive capsules on the terminal filament get smaller. Reading it backward, you can look at a tentillum under a microscope and make a decent prediction about the diet, which makes sense once you think about the mechanical problem. A copepod needs to be stopped and held. A fish needs to be penetrated, because a fish has scales and a mucus layer and enough muscle to tear free of anything that's merely stuck to its outside. The evolutionary sequence behind those parts is worth a moment, because it's a clean example of a structure being built up in stages. The Cystodonts, the Portuguese man-o'-war group, use penetrating capsules of a fairly simple type. The Siphonophores, which is everybody else, evolved the heteronemes. Then, on the branch leading to the group called Euclidophora, three things appear together: the segregation of penetrating and entangling capsules into separate regions of the tentillum, the terminal filament, and the collagen strand. Later still, in the group called Tendiculophora, the adhesive capsules on the terminal filament appear. And that last innovation is the one that opened the door to copepods. Before the adhesive trigger existed, the tentillum had no way to grab a small fast crustacean and hold it for the fraction of a second required to bring the main weapon to bear. After it existed, the most abundant food source in the ocean became available. So which siphonophores eat what? For a long time that was mostly guesswork, because watching one feed requires being in the right place with a submersible, and because a lot of what they eat is soft and disappears quickly in a gut. The workaround was genetic. Damian Serrano and colleagues published a metabarcoding study in which they sequenced gut contents from 159 specimens across 41 species, looking for fragments of prey DNA. 24 species produced identifiable prey sequences, yielding 55 distinct prey items in total. The overall picture was crustacean-dominated, as expected, with copepods accounting for the largest share. But the details are where it gets interesting. Closely related species turned out to be eating different things. Within the genus Bargmania, deep-living Physonects, one species had euphausiids and ostracods in its gut, another had mysids and copepods, and a third had ctenophores and copepods. Same genus, same general depth range, 3 different menus. That's the signature of competitive niche partitioning. Species dividing up a shared resource pool rather than all piling onto the same prey. Within Nanomaya, the partitioning ran by depth. The deeper living populations were taking krill and larger crustaceans. The shallower ones were on copepods, and revealingly, they were being selective. Cladocerans, small crustaceans that were abundant in the same water, were being avoided or were escaping. Either way, it wasn't indiscriminate. Some of the results overturned assumptions that had been in the literature for decades. Deep-sea siphonophores had been characterized as large prey specialists, based mostly on what people had happened to see through a submersible viewport, which is a method biased toward big conspicuous meals. The DNA said they were eating plenty of small crustaceans too, and shallow water species, thought to be crustacean specialists, turned out to be taking gelatinous prey, including salps and larvaceans. That last one has real consequences for how the open ocean works. Salps and larvaceans are filter feeders. They eat things too small for almost anything else to eat, and for a long time the assumption was that this made them a bit of a dead end, a lot of biomass that mostly sank, rather than moving up the food chain. Finding them inside Siphonophores means there's a route from the very bottom of the food web to a large predator in one step. And then there's the number that made me rethink the whole group. In Monterey Bay, M. Barry's long-running video surveys identify Nanomya bejuga as one of the principal predators of krill in the bay, ranked against everything else out there that eats krill. Researchers there have made the point that the collective Nanomya population takes more krill than all the whales that gather in the bay to feed on the same resource. Whales are what everyone photographs. The animal doing comparable work is a transparent string a few inches long that almost nobody has heard of, hanging motionless a few hundred feet down with its tentacles spread waiting. The curtain works on one condition: something has to swim into it. In the upper few hundred feet of the ocean, that's a safe bet. There's food up there, and it's moving. And if you hold a net in the water long enough, the net fills. Go deeper, and that assumption weakens. Prey gets sparser with depth, the water gets colder, everything slows down, and a passive net starts having to wait a very long time between meals. So a few lineages stopped waiting and started advertising. In July of 2005, Stephen Haddock, Casey Dunn, Philip Pugh, and Christine Schnitzler published a short paper in Science about a siphonophore in the genus Irena, collected by Mbari's remotely operated vehicles from between about a mile and 1.5 miles beneath the surface off California. Irena's tentilla are modified. Instead of the standard coiled nidoband arrangement, the ends carry structures that flick and twitch, and those structures glow. They produce their own light, and they also contain a red fluorescent material that shifts the wavelength of the light coming out of them. Red. That's the part that made the paper. Almost all bioluminescence in the deep ocean is blue-green, somewhere around the wavelengths that travel best through seawater. And the eyes of deep-sea animals have overwhelmingly evolved to match, tuned tight to blue, which means red light in the deep sea is close to invisible to most of the animals living in it. There are a handful of famous exceptions: dragonfishes that produce red light and carry the visual pigments to see it, and the standard interpretation of those is that red gives them a private channel, a searchlight their prey can't detect. So finding a siphonophore that produces red light and appears to be using it as a lure raises an uncomfortable implication. If the light is functioning as a lure, then something down there has to be able to see it. Haddock's argument was that red-sensitive vision among deep-sea fishes may be considerably more common than the textbook picture allows. That was the first time a bioluminescent lure had been documented in any invertebrate at all. Anglerfish had the concept locked up in the popular imagination. Nobody had caught an invertebrate doing it. And when they examined the gut contents of these animals, they found fish, which is consistent with the lure doing what it appears to be doing, though I'd note that consistency isn't proof of mechanism, and observing the actual moment of a fish being drawn in and taken at that depth is a very tall order. The animal was left unnamed in that paper. Over a decade later, in 2016, Pugh and Haddock published a formal description, and it went into the literature as Irena sirena. Sirena. Somebody in that room had a sense of humor about lures and songs and sailors. Now, the developmental point, because this is where the chronological thread we've been following pays off. That lure is a tentillum. It's a modified branch on a tentacle attached to the base of a feeding polyp, and that feeding polyp came out of a probud at a growth zone like everything else. Evolution didn't invent a new organ, to hold the light. It took a module that already existed, one that was already being mass-produced by an existing pipeline in fixed positions along the body, and edited what that module builds. That's the recurring theme in this group, and I think it's the actual explanation for why Siphonophores got so weird. The ProBud subdivision system hands evolution a set of interchangeable, independently modifiable parts, all coming off the same line. Change what one part becomes, and you've changed the animal, without having to renegotiate anything else. Delete a mouth, and you get a palpon. Enlarge a tentacle, and you get the Man-O-War's fishing zooid. Put a light on the end of a tentillum, and you get an angler. There are other variations worth mentioning, mostly because the deep-sea versions of these animals are so far from the popular picture. Bathyphysa conifera is a cysto-necked, so no swimming bells, and it looks like a tangle of pale tissue drifting in the dark. Deepwater ROV crews working on offshore operations started encountering it, and somebody nicknamed it the Flying Spaghetti Monster, which is now the name it travels under in a lot of the footage. It's a real, described species that's been known to science for a long time. It just happens to look like a joke. Then there's Maris. Deep-living physonects, several species, and they show up in the record of how hard these animals are to study. When Casey Dunn, Philip Pugh, and Stephen Haddock described Maris claudiniellus in 2005, they noted something you don't often see in a species description. Every single specimen they observed came apart. Some of them started shedding pieces when the submersible approached, before anything touched them. Others came apart during collection, or in the sample canister on the way up. The stem contracts hard, and parts of the animal simply let go. That's called autotomy, and it's the same category of behavior as a lizard dropping its tail. I'll come back to it, because it turns out to matter enormously for the question we're heading toward. Now let me go all the way to the other end of the group's range of solutions and talk about the one siphonophore almost everybody has already met. The Portuguese man-o-war is a cysto-necked. No swimming bells, no jets, no vertical migration, no dark. It lives at the surface, permanently, and its float has been enlarged into something no other siphonophore has. A gas-filled bladder that sits above the waterline, with a crest running along the top of it. A sail. The animal has no propulsion of its own. It goes where the wind takes it, and it has evolved a shape that means the wind takes it somewhere specific. The float is asymmetric. The crest is set at an angle relative to the long axis, and the tentacles trailing below act as a sea anchor, dragging in the water and resisting sideways movement. Put a sail at an angle above a keel, and you don't drift downwind. You sail across the wind. And the asymmetry comes in two mirror image versions. Some individuals are left-handed, and some are right-handed, and the handedness determines which side of the downwind direction the animal tracks. One form angles off to the right, the other to the left. Measurements of drift behavior have found the angle running to about 40 degrees off downwind in light conditions, and decreasing as wind speed rises, until in strong wind, the animal is being pushed close to straight downwind, regardless of which form it is. Why would a population maintain both mirror images? The leading suggestion is that it stops the whole population sharing one fate. If every man-o-war in the Atlantic sailed the same way relative to the wind, then a persistent onshore wind would put all of them on the same beach. With two forms, half the population goes one way. It's also been suggested that it helps avoid getting trapped in the same drifting windrows of sargassum and debris, where the wind concentrates floating material. Neither explanation is settled. But the dimorphism itself is well documented, and it's been known since the 1950s, and it's one of the few examples I know of an animal where left and right are separate life strategies. And then, last year, the man-o-war turned out to be several animals in a different sense than the one we've been discussing. A population genomic study published in Current Biology sequenced whole genomes from 151 specimens collected around the world. What came out was 5 distinct genetic lineages, with multiple independent lines of evidence pointing to real reproductive isolation between them, despite their ranges overlapping in the open ocean. That's a surprise on its own terms. The prevailing assumption about surface-drifting ocean animals with no barriers between them was that they'd be one big, well-mixed population, because what could possibly separate them? Wind and current move everything everywhere. Apparently, something separates them anyway. The team then did something I like a lot. They went to iNaturalist, the citizen science platform where people upload photographs of things they've found, scored thousands of images of man-o-wars from beaches around the world, and identified 4 recognizable morphologies. 4 of the genetic lineages matched 4 of those photographic forms. Several of those forms had been proposed as separate species back in the 18th and 19th centuries, then dismissed as variation within a single widespread species, and had been sitting in the discard pile ever since. The genomes brought them back. The recognized names now include Physalia physalis, Physalia utriculus, and Physalia megalista, and a newly described one, Physalia minuta, found around New Zealand and Australia. So the single most famous siphonophore on Earth, an animal that washes up on tourist beaches by the thousand, that closes swimming areas, that everybody has been photographing for 200 years, was at least 4 different species the entire time, and nobody could tell. While we're here, the practical part, and I'm going to keep this simple because partial advice about stings is worse than none. A man-o-war remains dangerous when it's stranded on the sand, and it remains dangerous when it's dead. Detached tentacle fragments in the water or on the beach can still fire, and stinging capsules don't need the animal to be alive to work, since the trigger is mechanical and the energy was stored when the capsule was built. Don't handle them. Keep children and dogs away from them, and follow whatever your local lifeguards and public health authority tell you, because first aid recommendations for jellyfish stings vary by region and by species, and the wrong intervention can make a sting worse. What strikes me about Physalia, in the context of everything we've been building up, is how far it went in the opposite direction from Arena. Same ancestry, same developmental machinery, same growth zone budding out chormidia in sequence. One lineage took that machinery down to 1.5 miles deep, turned off the lights, and put a red lamp on the end of a fishing line. The other took it to the surface, deleted every swimming bell it had, inflated its float into a sail, evolved left and right-handed forms, and handed navigation to the weather. Before we go any further, there's a piece of the life cycle I've been putting off, and it's the piece that closes the loop on everything the growth zone has been doing. Sooner or later, the colony has to make another colony. In a physonect like Nanomia, the gonophores develop inside the chormidia, budded off the same probuds as everything else, carried backward down the stem with their neighbors, maturing as they go. Male and female gonophores can occur on the same colony, which means a single animal is producing both kinds of gamete out of clonal tissue. When they're ready, the gametes go into the water, and everything after that is chemistry and luck. The Portuguese man-o-war does it differently, and the structure it uses is one of the more baroque things in the group. Hanging beneath the float, Physalia carries branched clusters called gonodendra. A gonodendron is a whole assemblage, gonophores in bunches, plus accessory zooids of several types clustered around them, including small polyps and structures that resemble reduced swimming bells, even though the animal has no swimming bells anywhere else. It's a reproductive apparatus built out of the same modular parts as the rest of the body, arranged into a subassembly with its own internal division of labor. There are reports going back a long way that mature gonodendra detach from the colony and sink, releasing gametes at depth rather than at the surface. That would make a certain amount of sense, since the surface is a hostile place to leave a fertilized egg, and the deeper water is where the rest of the group lives. I want to be straight about the evidence, though. Direct observation of Phasalia spawning is thin. This is an animal that everybody has seen and almost nobody has watched reproduce, which is a recurring pattern with these creatures and a useful reminder that familiarity and knowledge aren't the same thing. How long does any of this take? How long does a colony live? For most species, the honest answer is that we don't know. There are no long-term captive populations to age, and you can't tag a siphonophore. What we do have is population data, and the best of it comes from Monterey Bay, where Mbari has been running video transects through the same water column for decades. Nanomia bijuga abundance there rises and falls on a seasonal cycle, and the timing of that cycle is informative. The population peaks roughly 3 months after the onset of seasonal upwelling, and 1 to 2 months after krill numbers hit their own peak. That lag is the whole life history in one number. Upwelling brings nutrients up, which feeds phytoplankton, which feeds krill, which feeds nanomia. And the siphonophore population takes a month or two to convert an abundance of krill into an abundance of siphonophore. That's the time it takes for colonies to grow, since growth in these animals is a direct function of food, and for the resulting eudoxids and gametes and new colonies to work through. It also tells you something about the ceiling. A siphonophore has no fixed adult size, so an unusually good feeding year doesn't just produce more colonies, it produces longer ones. Abundance and body length respond to the same input. In a lean year, you get fewer, shorter animals. In a rich one, more and bigger. Now, who eats them? Less is known about this than you'd like, mostly because a fish that ate a siphonophore has almost nothing recognizable in its stomach an hour later. Gelatinous tissue digests fast and leaves nothing to identify. That's a general blind spot in ocean food web studies, and it's the reason gelatinous animals were written off as a nutritional dead end for so long, which turned out to be badly wrong once DNA methods arrived, and people started finding jelly in the guts of everything from tuna to penguins. For Physalia specifically, we have names because it lives at the surface where things can be watched. There's a sea snail called Janthena janthena that spends its life floating upside down beneath a raft of mucus bubbles it builds itself, drifting around the open ocean. And what it eats is the Portuguese man-o'-war. A snail with no ability to swim, hanging under a homemade float, eating an animal armed with some of the most effective stinging cells in the sea. And there's Glaucus atlanticus, the blue dragon, a nudibranch about an inch long, also floating at the surface, also feeding on Physalia. It doesn't merely survive the stinging cells, it collects them. Undischarged nematocysts from its prey are moved through its own tissues and stored in the tips of its appendages, where they're deployed against anything that tries to eat the nudibranch. So the weapons that a siphonophore builds in the basigaster of a feeding polyp, using a probud that came off a horn at the front of a growing stem, can end up being fired by an entirely different animal in a different phylum that swallowed them on purpose. I find that a satisfying place for a food chain to end up. Let's go back to that canyon off Western Australia, because now we have the vocabulary to describe what Sebastian was actually looking at. The expedition was called the Ningaloo Canyons Expedition, running in March of 2020 off the research vessel Falkor, with Nerida Wilson of the Western Australian Museum as chief scientist and Lisa Kirkendale as co-investigator. 20 dives, around 180 hours of exploration, working down to depths of nearly 3 miles. They came back with something like 30 species new to science, including glass sponges, a long-tailed sea cucumber, and a bioluminescent squid, plus a long list of first records for Western Australia. And they came back with the footage. The animal was at roughly 2,070 feet, and it was in a feeding posture, not swimming, not drifting slack, coiled into a spiral, holding the coil open, rotating very slowly. That posture is doing a specific job. A siphonophore's hunting surface is its tentacles, and the tentacles hang off feeding polyps, distributed along a very long, very thin body. If you leave that body straight, your interception surface is a long, thin ribbon, and you've spread yourself across a huge distance with no depth to your net. Wind that same line into a spiral, and you've packed the whole length into a compact volume, with tentacles filling the space inside the coil from every direction. Anything that swims into the middle of that has to cross the curtain twice to get out. I want to say clearly that reading intent into the shape is interpretation. Nobody has run experiments on the hydrodynamics of a 300-foot siphonophore in a feeding coil, for reasons that will become obvious shortly. But the geometry is not subtle, and this feeding posture has been observed in apolemia repeatedly. They measured it with lasers, which is the standard way to get scale from ROV video. The outer ring of the spiral came in at around 154 feet. That's the measurement. That's the number with instruments behind it. The 390-foot figure that went around the world is the team's estimate of the total length of the animal, derived by working through the geometry of the whole coil rather than by measuring the body directly. It's a reasonable estimate, and I have no argument with it, but it is an estimate. And the way it got reported, often with the qualifier stripped off, is a small case study in how a careful number turns into a confident one on its way through the news. Even so, the measured ring alone is longer than a blue whale, which raises the comparison everybody wants to make, and it needs to be handled carefully because it involves two different physical quantities that get treated as one. Length and size are two different measurements, and the ocean does not owe us a single winner. A blue whale runs to about 100 feet and can weigh well over 100 tons. That apolemia was longer, and nobody weighed it, and nobody could have. But look at what it's made of. A thread of tissue you could close your hand around, most of it water, held in a jelly matrix, with the working parts distributed along it. Whatever it weighed, it was a minute fraction of a whale, and that isn't a slight against the animal. It's the whole point of the design. It occupies an enormous amount of space using almost no material. For completeness, the title of longest animal has other claimants. The bootlace worm, Lineus longissimus, has reported lengths in the same territory, though the most extreme figures come from stranded specimens of an animal that stretches considerably when handled, and specialists treat those numbers with caution. Praia dubia, another giant siphonophore, is routinely cited at something like 130 to 160 feet, and the review literature describes giant apolemyids exceeding 100 feet as an established fact rather than a curiosity. I'd rather not spend too long on the ranking, honestly, because the ranking hides the more interesting question. The interesting question is what it takes to be that long. Go back to how the animal grows. A probud forms at the horn. The stem elongates, the probud gets carried backward and subdivides into a cormidium. Repeat, indefinitely, for as long as the animal lives and eats. There is no adult size, no developmental endpoint at which the animal stops and switches to maintenance. Growth is indeterminate, and a length measurement on a siphonophore is really a measurement of how long that individual has been running the process, minus whatever it's lost along the way. Which means the 300-foot apolemia isn't a different kind of animal from a 6-inch nanomya in the way a whale is a different kind of animal from a mouse. It's the same process further along. And the reason it can go that far without hitting a wall is that adding length doesn't change any of the engineering. When a vertebrate doubles in length, everything about it has to be renegotiated at once. Bone strength scales with cross-sectional area while body weight scales with volume. So a doubled animal is 8 times heavier and only 4 times stronger, and its skeleton has to be redesigned. Heat production scales with mass, and heat loss scales with surface area, so thermal management changes. Blood has to be pushed further against more resistance. Every one of those is a hard constraint, and every one of them limits how large a body plan can get. A siphonophore adding 100 chormidia changes none of that. The individual zooids stay exactly the same size they always were. Each feeding polyp handles the same size of prey. Each swimming bell moves the same amount of water. There's no larger version of any component anywhere in the animal. Length is achieved purely by repetition of a unit that never has to scale. That's the trick. And it's the same trick a coral reef uses, or a stand of aspens, or a fungal mycelium. Modular organisms get around the limits on body size by refusing to have a body size. But there is one constraint that doesn't go away. And I think it's the most interesting unsolved question about giant siphonophores. Nutrient transport. Remember, there's no heart and no blood. Everything moves through the gastrovascular canals, driven by cilia and by contraction of the stem. That's a perfectly good system over a few inches. Over 300 feet, it's a serious problem. Material moved by ciliary currents travels slowly, and the distance from a feeding polyp at one end of that animal to a growth zone at the other is enormous, relative to any plausible transport speed. So how does the front of a 300-foot colony get fed? The most reasonable reading, and I want to flag that this is inference rather than an established result, is that it mostly doesn't have to. A colony that long has hundreds or thousands of mouths distributed along its entire length. Any given region is surrounded by its own local feeding polyps. Rather than running a single centralized economy across the whole animal, a giant siphonophore probably operates as a chain of largely self-supplying neighborhoods, with slow exchange between them and the growth zone being the one place that truly depends on a net inflow. If that's right, and I'd love to see it tested, then the reason siphonophores can get this long is partly that they never built a body that needs to be supplied from a center. They were modular before they were large, and being modular is what let them get large. How many zooids are we talking about? Nobody has counted. I've looked, and there's no published census of a giant apollomyid, which isn't surprising when you consider what counting would involve. But you can bracket it. Chormidia in these animals are packed along the stem at intervals of millimeters. Take that spacing, run it along 300 feet of stem, allow several zooids per chormidium, and you land somewhere in the tens of thousands to hundreds of thousands. Treat that as an order of magnitude sketch rather than a figure, because that's all it is. Tens of thousands of bodies, every one of them a clone of every other. Every one of them derived from a single fertilized egg that you couldn't see without a microscope. There's a practical footnote to all this that I find slightly grim. Apolemia has shown up in the fisheries literature. Mass occurrences of apolemia have been implicated in mortality events at salmon farms in northern Europe, where large numbers of the animals drift into net pens, and the fish can't get away from the stinging tissue. The mechanism isn't a giant predator eating salmon. It's fragments of siphonophore tissue in the water, in quantity, in an enclosure the fish cannot leave, which tells you something about how these animals travel and how easily they come apart and how effective the weaponry stays after the animal that built it is no longer intact, all of which sits on top of something that has quietly shaped everything humans know about this entire group. These animals are not merely delicate. They are so fragile that for most of the history of biology, we were studying them wrong, and we didn't know it. Here is the standard way marine biologists sampled the open ocean for well over a century. You tow a fine meshed net behind a ship, at depth, for a while. You haul it up. You empty the cod end into a jar of preservative, and you look at what's in there. That method built most of what we know about plankton. It works beautifully on copepods, on krill, on fish larvae, on anything with a cuticle or a skeleton or a scrap of structural integrity. Run a siphonophore through it, and you get soup. The animal is jelly and water, held together by tissue that is measured in cell layers rather than in millimeters, moving through a medium where nothing ever pushes on it hard. Put that in a net being dragged through the water at a couple of knots for 20 minutes, along with a few thousand other organisms, and the colony disintegrates. The swimming bells come off. The stem breaks into segments. The bracts detach, the tentacles shred, and here's the trap. The pieces survive. Not alive, but intact enough to look like objects. A bract preserved in formalin is a clean, distinctive, describable structure. So is a swimming bell. So is a detached chormidium. So for generations, people were pulling up jars full of siphonophore parts and describing them. They described them carefully and with enormous skill. And a lot of that work is still useful, because the parts really are diagnostic. Ernst Haeckel produced a monograph on siphonophores from the Challenger expedition material in the late 1800s. And the plates in it are extraordinary. Some of the most beautiful scientific illustration ever made. But a substantial amount of the early literature consists of descriptions of fragments, named as though they were animals, because nobody had ever seen the whole thing and there was no way to know which fragments belonged together. Detached bells got their own names. Bracts got their own names. And the detached reproductive stages, which I'm about to spend real time on, got names and genera of their own, entirely separate from the colonies that produced them. Matching those pieces back up has taken a very long time, and it is not finished. To give you a sense of how not finished, there's a paper from 2014 describing the detached reproductive stages of 2 species in the genus Lensia, and reviewing every known bract of that type in the genus, because the correspondence between those free-floating pieces and their parent colonies was still an open question in the 21st century. That's the situation. An entire animal group whose taxonomy had to be assembled from the wreckage by people who mostly never saw one whole. What changed it was getting into the water. Blue water diving is the technique of scuba diving in open ocean with no bottom in sight, tethered to a line so nobody gets disoriented and drifts off, hand collecting individual animals into jars. You approach the animal slowly. You bring the jar around it. You cap it. No net, no drag, no crushing. It sounds simple, and it's a genuine skill. And it made intact siphonophores available to science for the first time in a meaningful way. Then came submersibles and remotely operated vehicles, and specifically, the gentle sampling systems mounted on them. Embieri's vehicles Ventana and Tiburon, working the water column off Central California for decades, produced a stream of intact specimens and, just as importantly, a stream of video of these animals behaving normally, undisturbed, in the place where they live. A great many of the siphonophore species described in recent decades were collected that way, mostly by Philip Pugh, often working with Casey Dunn and Stephen Haddock. More recently, blackwater diving, night diving in deep open water with lights, mostly driven by underwater photographers, has started contributing real observations too. There's published work using Gulf Stream blackwater dives to document siphonophore diversity and behavior, which is a nice case of a hobbyist community feeding into a research field that's starved for encounters. Even with all of that, the totals stay small. A global review published in 2014 recognized 175 valid species in 16 families and 65 genera. 5 of those species are cystonyx, the man-o'-war group. Around 170 are calicophorans, and new ones keep arriving, which tells you the curve hasn't flattened. 175 species for an animal group present in every ocean, at every depth, in numbers large enough to be a principal predator of krill in a productive bay. Compare that to something like 40,000 described crustacean species, and you get a sense of the sampling problem. Now, some of this fragility is a research inconvenience, but a lot of it is a genuine property of the animal, and the difference matters. Maris claudanielis is the case that makes it undeniable. When Dunn, Pugh, and Haddock described the species in 2005, they recorded that all of the observed specimens shed parts of themselves. Not because the submersible hit them. In some cases, the animals started coming apart as the vehicle approached, before contact, presumably in response to the pressure wave or the lights or the sound. That's autotomy, voluntary shedding. The stem contracts violently and sections of the colony let go. A lizard drops its tail because a predator has hold of it, and the alternative is worse. A sea cucumber ejects its internal organs for the same reason. Autotomy is a trade, tissue for survival, and it only makes sense in an animal that can afford to lose the part. A siphonophore, apparently, can afford to lose a lot of parts. And I want to stay on that implication for a second rather than rush past it. If pieces of this animal detach in response to a threat, then the boundary of the animal is negotiable in real time. It has a policy about how much of itself to keep. That's not a property most bodies have. When something grabs you, your options do not include reducing your extent. There's a version of this that's a little unnerving to think about, and I don't have evidence for it, so I'll frame it as a question rather than a claim. When a colony sheds a section of stem carrying a dozen chlamydia, what happens to that section? It has mouths on it. It has stinging capsules that still work, since the capsules are mechanically triggered and don't require the animal to be alive. It has bracts to keep it buoyant. What it doesn't have is a growth zone, so it can't build anything new, and it can't swim, so it can't get anywhere. It drifts, and it stings, and it presumably starves eventually. Whether it feeds in the meantime, and for how long it persists, I have not been able to find good data on, which is a fair summary of the state of knowledge on most questions about these animals. There's a related and better documented consequence, though, and it's the salmon farm problem from a moment ago. When siphonophore tissue ends up in a net pen in quantity, the damage is done by fragments. Bits of stinging tissue in the water, in an enclosure the fish cannot swim out of. The weaponry keeps working after the animal has stopped being one, which is a strange sentence to write about a body. And it points at the last piece of this, which is the piece I've been building toward since the growth zone. Because so far I've been describing fragmentation as damage. Something happens to the animal and pieces come off. Bad luck, or a defensive trade, or a net. In one large group of siphonophores, fragmentation isn't damage at all. It's the plan. Follow a chormidium all the way to the end of its life and watch what happens. It buds at the horn as part of a probud. It subdivides. Its zooids differentiate. It gets carried backward down the stem as newer chormidia are inserted ahead of it. It matures. Its feeding polyp catches things. Its gonophore develops gametes. And it keeps moving backward because the growth zone never stops adding, and eventually it arrives at the very end of the animal, the terminal position, with nothing behind it. In most calicophoran siphonophores, that's when it leaves. The chormidium detaches from the colony, whole, all of its zooids together, still connected to each other, still sharing a gut, no longer connected to anything else, and it swims away. The detached unit is called a eudoxid, and it is, in my opinion, the single most interesting object in this entire group of animals. Here's what's in one: a bract, the gelatinous shield, which handles protection and buoyancy, a gastrozoid, complete with its mouth and its tentacle, and the tentilla on that tentacle, which handles feeding and prey capture, a gonophore, which handles reproduction, and in most species, a swimming bell of its own, giving it propulsion. Read that list again: feeding, defense, buoyancy, movement, reproduction. That's the complete function set of an animal. The eudoxid swims off into the plankton. It hunts on its own account. It grows its gametes. It spawns. It does everything an organism does, using organs that are, technically, separate individuals, in an arrangement that budded off the end of something much larger and left. And in the older literature, it was classified as its own animal, in its own genus, with its own name, entirely separate from the colony that made it. That's where the word comes from. Eudoxia was the genus assigned to these free-swimming things by people who had no idea what they were looking at. The term eudoxid is a fossil of a misidentification. Somebody netted a small transparent object with a shield and a stomach and a gonad, described it as a new animal, and they were wrong in the interesting sense rather than the careless one, because by every functional test they had available, it was one. Working out the correspondence between eudoxids and their parent colonies has been slow, tedious taxonomic work that in some genera is still going on. You essentially have to keep a colony alive long enough to watch it release something, then match the released thing against the free-swimming forms in the collections. The recent papers matching detached stages to species in Lensia exist because that job is not done. Now, the obvious question, is a eudoxid an escaped piece, or is it a stage in a life cycle? It's a stage. Absolutely, unambiguously, by design. And the evidence for that got a great deal better recently. A study published in Current Biology went at the mechanism directly, working with Chelophys appendiculata, a calicophoran, using live imaging, immunohistochemistry to map tissue architecture, and pharmacological inhibition to knock out specific functions and see what broke. The release of a eudoxid is controlled by a dedicated muscle. There's a specific muscle whose job is to detach the terminal cormidium from the colony. It isn't the stem contracting generally, and it isn't tissue tearing under load. There's a structure built for this, at that location, that performs a separation, and the release involves tissue remodeling, meaning the connection between colony and cormidium is actively reorganized rather than simply broken. The result, in the author's framing, is a physiologically integrated dispersive unit with its own behaviors. Their phylogenetic analysis suggested the whole arrangement evolved once, through a simultaneous reorganization of colony architecture and the evolution of the zooid structures that make the detached unit viable. It's a package. You don't get the eudoxid without also getting the changes to the stem and the changes to the zooids that let a fragment survive on its own. And there's an ecological detail in that study that I keep chewing on. Eudoxids and their parent colonies often have different distributions, different depths, different regions. They're not simply floating along near the parent doing the same thing in a smaller body. They occupy a different slice of the ocean, which means the species is running two different ecological strategies simultaneously with the same genome, in two different body configurations, at two different scales. Compare this to how the rest of the hydrozoans do it, because that comparison is where the significance lives. A typical colonial hydrozoan, growing on a rock or a piling, produces medusae. The medusa is a single zooid. It buds, it detaches, it swims off, it matures, it spawns. One body leaves. A siphonophore that produces eudoxids is doing the same thing one level up. What detaches is a small assemblage of several zooids, and that assemblage does the swimming and the feeding and the spawning together, as a unit. The dispersal stage changed level. What used to be an individual leaving a colony became a small colony leaving a bigger colony, and behaving like an individual while it does it. That's the transition the current biology authors were pointing at when they described the eudoxid as an elusive evolutionary transition in individuality. There aren't many places in biology where you get to watch that boundary being crossed by a specific, identifiable mechanism. Usually, these transitions are ancient, inferred, and reconstructed from comparative evidence. This one has a muscle. Now let me push on what the eudoxid actually tells us. The survival of a fragment is the easy half of it. The hard half is which fragments qualify. Take any other section of a siphonophore and detach it. A run of swimming bells from the nectosome, no mouth anywhere in it. It can swim beautifully, and it will never eat again. A bract. No mouth, no muscle, no nerves worth speaking of. And it will drift until it decays. A palpon. No mouth. A section of stem carrying gastrozoids, but no bells. It can eat, it cannot move, and it will sink or drift according to whatever bracts came with it. Every one of those fragments is missing something, and missing it permanently, because the growth zone stayed behind with the parent, and there are no interstitial stem cells in the stem to build a replacement. The eudoxid works for exactly one reason. It happens to contain one of each necessary thing. That's it. That's the whole criterion. Size has nothing to do with it, and neither does complexity, and neither does the amount of tissue that came away. The question of whether a detached piece of a siphonophore is an animal or a scrap comes down to whether the piece includes the complete set of functions, because none of the parts can generate a missing function from scratch. And the last core medium on the stem includes the complete set because that's where the probud subdivision put things, in that order, in that arrangement, at that position, weeks earlier, at a bulge of tissue several feet away at the other end of the body, which means the individuality of the eudoxid was determined by a budding pattern. Even so, the eudoxid is missing one thing, and it's the thing that makes it a very peculiar kind of individual. It has no growth zone. It can't add zooids. It can't extend. It can't repair a lost bract or replace a damaged feeding polyp. Whatever it left the parent colony with is what it has for the rest of its existence. It's a complete animal with no developmental future at all, running on a fixed inventory, doing one job before it stops, which is a strange sort of organism to be, fully functional, fully independent, ecologically distinct from its parent, and permanently incapable of building anything. I find myself thinking about what that means for the parent colony too. From the colony's point of view, if a colony can be said to have one, the eudoxids are its output. It spends its life converting captured copepods into chormidia, carrying those chormidia backward down its own body while they mature, and releasing them off the end. The colony is a production line whose product is small animals, and the product is made of the same tissue as the factory, and the factory got there by being a slightly larger version of the same thing. Somewhere in that description, the words stop working properly, and I think that's the honest place to end up, because we've now got a group of animals where a fragment of a colony is an individual, where the colony itself started as an individual, where each part of the colony is homologous to an individual, and where the deciding factor in every one of those cases is the same small set of developmental facts, which is where all of this has been going. So let's put the whole sequence in one line and see what it adds up to. An egg divides. The divisions sweep across it from one pole. A layer of endoderm forms preferentially on one side, and that side thickens, and the thickening decides where everything will be built. A polyp differentiates at the oral end. A float invaginates, or a swimming bell buds, depending on the lineage. The stem elongates. The budding region splits in two, and the two halves drift apart along the growing stem. Each of them develops a horn, and the interstitial stem cells concentrate there and nowhere else. Probuds form, get carried backward, subdivide into chormidia. The chormidia mature as they travel. In some lineages, they detach at the end. Every step in that sequence is a normal developmental event. Cell division, tissue folding, regional specification, budding, differentiation. Nothing in there is exotic. You could find every one of those processes in a sea anemone or a hydroid or a coral. And at the end of the sequence, you have an organism that biology cannot categorize. I've been circling this the whole way, so let me put it down plainly. There are two things that decide whether a mass of connected tissue counts as one animal or many. Only two, as far as I can tell, and both of them are settings in a developmental program rather than principles of any kind. The first is where budding stops. If the capacity to build new bodies is distributed through the organism, then any piece you break off carries some of that capacity with it, and any piece can potentially become a whole. That's a hydra. That's a hydroid colony, spreading across a rock. That's a stand of aspens, where every trunk is connected underground and any trunk could carry on alone. Those organisms fail to be individuals in a specific mechanical way. Their parts can regenerate the rest. If the capacity to build new bodies is confined to one location, then the pieces that break off are terminal. They can't become wholes. They finish whatever they already were, and they stop. That's a siphonophore, with everything concentrated at 2 horns of tissue, and no interstitial stem cells anywhere in the connecting stem. The second thing is how far the parts have gone in losing the ability to live alone. A coral polyp in a colony can feed itself, defend itself, and in many cases survive detachment. It's an integrated part of a larger structure, and it's also a functional animal. Both at once, no tension. A siphonophore swimming bell has no mouth. It never had one. Its development includes the standard beginning of a medusa, and then simply stops before the parts a free-living medusa needs. The gonophore stops earlier. The palpon has had its mouth deleted. The bract is a shield with plumbing. Every zooid in the colony has had something removed, and the removals are complementary. So the assemblage works, and none of the pieces do. Those 2 variables are what the argument has always been about, even when nobody was stating them that way. And notice that neither one is about counting bodies, or about genetics, or about how tightly things are stuck together. They're about developmental capacity and functional deletion. Both are measurable. Both are physical. And usually, in most organisms, the 2 variables agree with each other, which is why the word individual works fine almost everywhere, and nobody has to think about it. In siphonophores, they disagree, and they disagree in both directions at once. By the deletion test, a siphonophore colony is unambiguously one animal. Its parts cannot live alone. They're more dependent on each other than the organs in your body are on each other, in the sense that a heart in a machine can be kept beating, and a swimming bell in a jar cannot be kept doing anything. By the developmental origin test, it's unambiguously many. Each part traversed the developmental route of a free-living animal and stopped part way. They are, historically, individuals. Both tests are valid. They give opposite answers. And the reason they give opposite answers has nothing to do with philosophy. It's that this lineage happened to evolve extreme functional specialization while retaining the developmental machinery of colony formation. And those 2 things ordinarily don't travel together. Now do the same test on yourself, because this is the part that I think turns the question around. Your parts have gone further down the deletion path than a siphonophores have. Not one cell in your body could survive independently in the ocean. Your liver has no capacity for autonomous existence of any kind, and your ability to build new bodies is confined more tightly than a siphonophores. It's restricted to a germline that was set aside early in development and can produce exactly one thing: a gamete. By both criteria, you're more of an individual than a siphonophore is. You're further along both axes. The only reason nobody calls a human a colony is developmental history. Your organs never had the developmental root of free-living animals. They were always parts. They came into existence as regions of a single embryo, specified by position and signaling, with no history of independence to lose. A siphonophore's parts did have that history, and they still run the program, right up to the point where it stops. So the boundary between a colony and an individual comes down to that: where the budding is allowed to happen, how much has been deleted from each part, and what developmental route those parts took to get there. 3 facts about growth. That's the whole distinction, and it doesn't sit at any particular point on a scale, because organisms are scattered all over that space, rather than lining up along it, which means we're dealing with a region instead of a line. Organisms are scattered all through that space at different coordinates, and the ones that land in the awkward corners get argued about. Siphonophores sit in the most awkward corner anybody has found, and I don't think the appropriate response to that is to pick a side and defend it. I've read a fair amount of the literature on this, and the people closest to these animals mostly don't bother arguing the point anymore. They describe the mechanism, they specify the level they're talking about when they say something, and they move on. Dunn and Wagner made the case years ago that the productive question is how colony-level organization evolved in the first place, and what made this degree of integration possible, and that question has actual answers in it. The honest position, I think, is that individual was never a natural category. It was a word we generalized from the animals nearest to us, most of which developed from an embryo into a single body with no budding stage anywhere in the process. That covers vertebrates, arthropods, mollusks, most of what a person encounters in a lifetime. And then we discovered that a large fraction of life on this planet is modular, and the word started producing nonsense at the edges. There's still a great deal we don't know here, and I'd rather leave you with the gaps than with a tidy conclusion. Nobody has identified the molecular signal that establishes the dorsal ventral axis in a siphonophore embryo, which is the very first event that determines where the budding will occur. Nobody knows what confines the interstitial stem cells to the horn, or what would happen if that confinement failed. Nobody has counted the zooids in a giant colony. Nobody has watched a lure work at depth from beginning to end. And the reason for most of those gaps is the same reason the taxonomy was built from wreckage. These animals come apart when you look at them wrong, and until very recently, nobody could keep one alive long enough to run an experiment. That's improving now. Culturing methods have gotten better in the last few years, which is exactly why the developmental work I've been quoting is so recent. So here's where I've landed, and it's a smaller claim than it sounds. A siphonophore is a body whose parts kept the developmental history of separate animals while losing the ability to be them. The boundary between colony and individual, in this group, is set by where the growth zone sits and how much each part had removed. Both of those were fixed by a budding pattern in the first days of the animal's life, for reasons unrelated to the question. The line between one and many turned out to be an accident of development, and the accident happened early, and nothing since has been able to undo it. That's the thing I'd want somebody to take away from this. A question that seemed like it should have a clean, obvious answer turns out to be settled by the position of a bulge of tissue at the front of a stem, and by how much each part had taken out of it on the way to the job it does now. If you've stayed with me this far, do me a favor and settle something. Somewhere in the sequence I just walked through, the count changed. It went from one to many, or from many to one, depending on which direction you think it runs. And I'd like to know where you put it. Was it the moment the first bud appeared on the ventral endoderm? The moment the swimming bell finished developing without a mouth? The moment the growth zones split in two? Or do you think the count never changed at all? And it was one animal the whole way through, at every stage, no matter how many mouths it ended up carrying? Put your answer in the comments and be specific about the step, because the specific step is the interesting part. I go through the comments on these. And the disagreements down there are usually better reasoned than the paper abstracts. And if you want more of this kind of thing, the kind where an ordinary question turns out to be resting on a mechanism nobody mentions, subscribe and hit the like button, because that's the sort of small signal that tells me which of these are landing. Thanks for watching. Wherever you decided to draw that line, at least now you know what you were actually deciding about.